Molecular imaging is rapidly gaining acceptance as a clinical diagnostic tool.
Molecular imaging is revolutionizing medicine. When a person goes to a doctor, the doctor identifies what the problem is, asks the question, where in the body is the problem, what's going to happen to the patient, and what can be done about it. That is treatment. We have gone from considering only the symptoms and the physical examination abnormalities to the study of organs with conventional x-rays. We've gone to cells as examined by histology and pathology, but today we're moving and are in the molecular world where we identify abnormalities in the molecular processes going on in the body. Molecules provide the energy for life, they control communication between cells, and we can now measure these outside the body. Blood tests could be thought of as molecular imaging, which goes back for a long time, but today we can measure in various parts of the body what we call in situ imaging of these molecular processes, and they're the targets for treatment and diagnosis.
Image optimization requires a marriage among the medical camera, software, and diagnostic radiopharmaceutical agent.
In 2003, Philips selected CellPoint as a collaborative partner because of the mutual belief that EC technology could optimize imaging in both cardiology and oncology applications. It was mutually decided that there was a requirement to develop unique software to allow the optimization of the imaging. The software that was developed by Philips is software that integrates the imaging characteristics of the camera with the biological distribution of the imaging agent. The resulting product, referred to as Astonis, has the ability to improve image quality as well as to quantify the aggressiveness of tumors and to determine the most appropriate response. The overall agent development with the software has allowed Philips to become the leader in the development of software technology for molecular imaging.
When cancer cells grow out of control, they gobble up nutrients in the bloodstream faster than any other cell. It's why cancer tumors are so dangerous, but it may also be their biggest weakness. Terry Collip is one of the founders of a company called CellPoint. His product is almost like science fiction. It's a special compound made of sugar, the kind eaten by cancer cells, and a radioactive isotope called technetium-99. Injected into the body, cancer cells grab on and hang on.
Now, when that's injected into the bloodstream, it's the cancer cells that take in the glucosamine. And when it does that, it's like a Trojan horse. The technetium goes in there, and the camera sees the technetium, of which it can go ahead and light it up.
In other words, even tiny pockets of cancer light up like a bonfire. Now, what makes this process so novel is that it doesn't require any expensive new scanning devices or cameras. It uses spec cameras already in most hospitals and medical clinics across the world. Universities doing clinical trials of the new compound are finding that it only binds to cancer cells and not inflamed tissue. That means fewer false positives, fewer wrong diagnoses, fewer surgeries.
An exciting new weapon in the fight against cancer. It's a safer, cheaper way of pinpointing cancer cells, and it looks like it's about to win approval for use in hospitals and clinics nationwide. The technology uses a new chemical compound that makes cancer cells sort of light up, makes them easier to spot with cameras that most hospitals already have, saving them and you a lot of money. Dr. Mark Siegel is a Fox News medical contributor. In this case, you put this radioactive isotope in a sugar solution, the cancer cell eats it, and then goes off into the body, right?
John, that is a great analogy. It's what they call a molecular glue, where you take a tendency that the cancer cell has and you put it together with an imaging technique to actually find the cancer cell. The reason that that's a great improvement is right now we're fumbling along looking for inflammations in the body. We have PET scanners. We have very up-to-date techniques for finding inflammation, but it can't always separate out cancer from non-cancer inflammation. This would be able to do that because cancer itself has certain tendencies, like it loves to eat sugar.
All right, so this radioactive isotope that they give you, this is nothing that's going to be, long-term dangerous, I guess?
No, really, because it's using the same isotope we already use. We use technetium already in our studies, nuclear imaging, and it's been very, very safe. It's a very low-grade radiation, and it really hasn't shown to be a problem at all. It may also be a way to deliver therapy to cancer cells. First you find it, then you deliver a poison and get rid of it before it spreads.
Dr. Mark Siegel, a Fox News medical contributor.
Thank you, John.
Promising. Thank you.